Hello, everyone. I'm Zuofu, a senior from Macalester College. Today, I'm excited to talk about my summer 2020 research with Dr. Kelsey Greendy on statistical methods for studying human genetics among people with mixed ancestry. In this presentation, I will be focusing on using Rcpp to speed up a tool for controlling for multiple testing in genetic data. Our research is built on the background of genome-wide association studies, which is a method for identifying the associations between certain genetic regions called loci and one's distinguishing characteristics called traits. We have seen a lot of studies using GWAS to identify the differential risk by loci, such as the study of breast cancer that was published on Nature Communications 2019. However, in traditional genetic association studies, people with mixed ancestry, such as African Americans and Hispanic Latinos, are disproportionately underrepresented. To understand more about differential risk brought by different ancestries, a concept called admixture mapping comes in handy, which is a gene mapping method that studies a population of mixed ancestry, like what we see on the right. In generation 3, we see this almost mosaic pattern from mixed ancestry. We can then find the loci contributing to the traits or diseases in this population. Suppose we have a person with two mixed ancestry, H and M, and we have multiple loci that determine their eye color, like what we see on the table. At each locus, we record how many copies you get from each ancestry. The two columns add up to 2 because you get one from the father, one from the mother. Many observations like this form a data set. Since the two columns are perfectly correlated, we can shrink the two columns down into one. And eventually, we may end up with a matrix on the right-hand side. Next, we perform marginal regression, like what we see in the formula. Aij refers to the number of alleles inherited by individual I at locus j. Alpha is the intercept, and pi is a vector of confounders. We then step along the genome and perform hypothesis tests locus by locus. We have a total of m hypothesis tests. And then the issue of multiple testing will arise. Suppose that the testing threshold we have is 0.05, which means the probability of type 1 error will be 0.05. Suppose we have 10,000 hypothesis tests in total. And then the family-wise error rate, which is the probability of making at least one type 1 error across all the tests, goes to 1. Given the high-dimensional nature of this data, m is usually on the order much higher than 10,000. And this number may be even larger if we have more than 2 ancestries, which means we need to use smaller significance thresholds in order to control the family-wise error rate at a reasonable level. With traditional multiple testing techniques, we often land at conservative results because they do not take into account the correlation between tests. This is more so in genetic studies because of how DNA is inherited. Neighboring loci are highly correlated. My work this summer is based on the new approach of getting the multiple testing significance threshold introduced in my advisor Dr. Greendy's paper. The threshold was based on the asymptotic joint distribution of the admixture mapping test statistic. The results are implemented in the STEAM package. In this package, correlation between test statistics are explicitly modeled. My job is to speed up the computational pipeline.
First, what is Rcpp? Rcpp is a package in R that binds the power of R and the efficiency of C++. We first identify the places where the existing package could be slow, such as the for loops that the interpreted languages such as R are not very good at. Then we implement the related functions in C++ code. The most important step is to test whether these improvements actually work. It's tricky to predetermine which code are best in C++ and which should be kept in R. So it's important to test different levels of Rcpp implementation. Finally, we found the most efficient approach and implement them in the version we have. We note that many steps are still done in R because many R functions were optimized in efficiency, and transforming these codes to C++ may not bring big benefits. After we transfer core steps from STEAM to STEAM CPP, such as the step where we simulate the test statistic, we record the time for obtaining the significance threshold. When the number of ancestries is 2, the time for obtaining the threshold reduced by approximately 75%. Benchmarking shows that our approach is even faster when the number of ancestries increases. And there's some future work that I would like to discuss. For example, we would like to introduce STEAM CPP in a broader setting. Currently, the working version is published on GitHub. You can use the QR code to access a mirror repository. Our goal is to clean some messages, add vignettes, and potentially publish to CRAN or Bioconductor. Finally, here are some references for this presentation.